Welcome to the Introduction to Biopharmaceutics and Pharmacokinetics. The purpose of this lecture is to introduce the main topics of the entire course. So even though we're going to be talking quite a bit about biopharmaceutics, this is just the introduction. So I will have a full lecture on that, and then many lectures on pharmacokinetics. I will be talking about the basic pharmacokinetics in my lectures, and then the remaining clinical lectures will be on the clinical pharmacokinetics, which will incorporate a lot more discussion of different medications that are relevant, that are most important in pharmacokinetics. Learning objectives for this lecture are to define drug product performance in biopharmaceutics, describe how biopharmaceutics affects drug product performance, define pharmacokinetics, and describe how pharmacokinetics is related to pharmacodynamics. And if you don't know what pharmacodynamics means yet, you'll find out fairly soon, and drug toxicity. Define the term clinical pharmacokinetics and explain how clinical pharmacokinetics may be used to develop dosage regimens for drugs in patients. Define pharmacokinetic model and list the assumptions that are used in developing a pharmacokinetic model. Okay, so when I say the word model, it's generally going to just be a few simple boxes with a few things called kinetic constants or rate constants. And the whole purpose of a model is to give you an idea of the way you want to kind of mathematically represent what is happening in the body with the drug. So there are simple models and there are complex models. I'm just going to introduce the idea of a model, just what it looks like in general. There won't be many calculations for this lecture, but as we proceed, once we get into actual pharmacokinetic lectures, of course, we'll have a lot more calculations. In the last bullet, it's not something that you'll necessarily be tested on, but I will describe or show where you'll find this kind of information, this pharmacokinetic information when you're doing, when you're looking at a package insert. Where do you find pharmacokinetics and how do you use that information when you're actually looking at a drug monograph or package insert for a medication? So drug product performance is actually a specific term used in biopharmaceutics. So a variety of dosage forms exist: tablets, capsules, ointments, creams, liquid suspensions, emulsions, and many other dosage forms that you've probably seen already. A drug product is a drug delivery system that releases and delivers a drug to the site of action. So, so the active ingredient is one thing, but a drug is made up of a lot of other things. Okay, and when you combine whatever else is in the drug with the active ingredient all together, that's going to be the whole drug product, right? The drug products must meet the individual patient's needs. Palatability means, of course, can they tolerate consuming it or swallowing it, or whatever method you're administering it? Can they handle that? Is it bearable? Is it comfortable enough? Convenience and safety. Okay, drug product performance is the release of a drug substance from the drug product either for local or systemic action. So that's specifically saying, is the active ingredient being released in the appropriate way, the appropriate rate, appropriate extent, in the right location. So it's really specifically about the drug being released from the product. So it's not a very vague, even though it might look vague, as a specific definition. So biopharmaceutics is the interrelationship of physical, chemical, which if you put that together, you can say physicochemical as one word. So physicochemical properties of a drug, dosage form, route of administration, and how those affect the rate and extent of drug absorption. So in simple words, in my own words, biopharmaceutics would be basically, all the properties that determine that drug's absorption, actually disintegration, dissolution, and absorption. It's in the whole grand scheme of a drug's pathway from beginning to end in the body, biopharmaceutics is the beginning. How is the drug breaking down? What are the factors that affect its disintegration and dissolution, etc. So if this is the grand scheme here, and this is almost everything in kind of a nutshell, what happens with the whole drug from the beginning to the end. And remember, the end is going to be either excretion. Well, it's always going to be excretion, but at some point we'll also have a pharmacological or clinical effect. And then in between that, there's distribution, right? There's drug in the systemic circulation or drug in the tissues, or it'll bounce back and forth. But everything in the beginning here is really related to biopharmaceutics, okay? And that might not be something that is intuitive based on the word, but if you think of pharmaceutics, pharmaceutics is generally drug formulation, and bio is basically how your biology interacts with the drug formulation. Okay, so that's kind of why it's called that. So in biopharmaceutics, the sequence of events are drug administration, release from the dosage form, absorption, and then hopefully a pharmacodynamic effect. That means the drug is having some action on its intended target. Okay, and then drug metabolism can occur before or after the pharmacodynamic effect, and may be required for effectiveness and or elimination. So that's a lot to say in one statement, and that last statement means drugs can be chemically modified in the body, right? That's called metabolism. That can happen any time, right? That could happen before the drug is active, or it could happen after the drug is active, and it could deactivate it. Metabolism could produce metabolites that are that still have activity. So when it comes to metabolism, the safest bet is to not assume anything, right? In terms of what will happen to that drug after it's metabolized, unless you know ahead of time. So metabolism can occur almost any time once the drug is, even when it's just starting to get absorbed, right? Just in the maybe if you're having an oral dosage form in the small intestine, you could actually have some metabolism even before you get to the liver. So that could be anywhere, but biopharmaceutics is mostly the drug release and absorption. The drug product formulation could have a substantial effect on drug absorption, even at the same dose and route of administration. That means if I have the exact same amount of active ingredient, I'm giving it the exact same way. It could be oral, for example, the most common way, an oral administration of a medication. Just because I have the same active ingredient, the same amount of active ingredient, doesn't mean it's going to result in the same outcome, because that depends on how the drug is formulated. It's really important that the drug is formulated in a consistent manner. So much excipient. Does it have enteric coating? Do we have to stabilize it from stomach acid? There's so many things that can happen that we need to control. This is why generic drugs cannot go on the market without FDA approval. Right? It is very true that generic drugs are, in almost every case, they're going to be very, mimicking the brand name very, very well in almost any case. And there might be a few exceptions for patients that might need the brand name, but for the most part, generics, the regulation is so kind of high on that in terms of its kind of replicating the biopharmaceutics that basically generics are pretty much equivalent. A brand name drug developer does not have to make their formulation method public, meaning the dosage form is not simple to replicate. So that might conflict with what I just said. So let me clarify. That means, the drug developer that first made the drug, it's the first time the drug has been on the market, they don't have to disclose their method of formulating the drug. They don't have to tell you exactly how they made it, and their excipients or the inactive ingredients, they have to be safe, but they don't have to tell you every detail, and it doesn't have to be in public. So how does a generic company go about replicating that? Well, the only thing they have to absolutely replicate is the drug absorption profile. Right? It has to be the same active ingredient. It has to be absorbed at the same rate and extent. Right? And that's actually, believe it or not, that's one of the last lectures of the whole course for my section at least. The last lectures, one of the last ones is bioavailability, and that's what we're talking about here. So the main point is they have to be absorbed to the same extent, the same rate, and that is going to depend on how fast they get dissolved in the first place, right? The tablets or capsules have to dissolve to a similar extent, in a similar rate, brand name and generic, in order to be equivalent. And that's called bioequivalence. Bioequivalence specifically refers to, again, the absorption profile of a drug. We'll talk about that much later on, like, as I said. The approval process is run by the FDA Center for Drug Evaluation and Research, CDER. In fact, I know of a few PharmDs who ended up working at CDER, and I was offered a job at CDER because they like to get pharmacists and sometimes PhDs to evaluate drugs for bioequivalence, and that can be a full-time job, for some professionals there. So that's called CDER or CDER. There's an Office of Generic Drugs specifically, and they would approve the abbreviated new drug applications, which would be just for generics, or new drug applications would be a different office, not the Office for Generic Drugs. Okay, so again, that's all regulated by the FDA. So bioavailability or bioequivalency is not something we just hope for the best and we all hope it happens. We have to prove, these companies have to prove that they have bioequivalence with their generic. So what are the main things that we consider in biopharmaceutics in drug product design? Okay, there are, I'm not going to go over every detail of each one of these, and so I'll break some of these down later that I consider more relevant. But there are critical manufacturing variables. It could be something as simple as what's the temperature of manufacture, how long do you grind it for when you grind up the powder before you put it in a tablet. It could be anything you could think of in the climate control, humidity, anything. That has to be controlled, believe it or not. All that has to be controlled. Therapeutic objective. Of course, when you're developing a drug formulation, you need to understand where the drug is supposed to target, where it's supposed to go, and what it's supposed to do. Particularly the target, what tissues it's supposed to go to at a high enough concentration. Active pharmaceutical ingredient. Well, the active pharmaceutical ingredient or the actual drug, when you say, omeprazole or esomeprazole or any active ingredient that's called the API or active pharmaceutical ingredient. That's what's actually doing what the actual therapeutic thing inside the body. Okay, but that API it's a chemical, right? So it has certain, in some cases it might be labile or sensitive to certain environments, and you have to treat that very carefully and know that you can mix it with this, but you can't mix it with that. And essentially, you have to think about the API when you're thinking about the whole formulation. The route of administration, of course, you don't normally see tablets going in a rectal route, right? You would see a suppository. So that's just an obvious one, but some of these might not be as obvious that we'll get to. But route of administration will dictate the type of formulation you're going to produce. Drug dosage and regimen. Yeah, so if you want someone to take something once a day, you might change the formulation. Whereas if it's okay to administer it 2 times or 3 times a day, you might rely on a different formulation. So your formulation is going to dictate. Well, actually, the convenience and patient factors will dictate basically how we want to formulate that drug in order to have kind of an adequate and a regimen that's convenient and adequate. Dosage form. And these are all kind of like interdependent. So like I said, dosage form will depend. That'll depend on your intended target, right? Where do you want the drug to go, and how are you going to administer it? That'll determine, is it going to be a tablet, is it going to be a capsule, is it going to be a gel, is it going to be a lotion, if it's topical. These are all something to consider. Excipients are the inactive ingredients, and as I used to bust Dr. Kibbe, which he used to teach. He's an emeritus professor. I used to tell him that those are the parts of the drug that don't really do anything. In reality, they're important because they determine, in many cases, they'll determine how the drug how fast it's disintegrating and how fast it dissolves, how fast it's absorbed. The excipients can have a major effect, and even on drug stability, the excipients can have an effect. So I already mentioned, kind of off the cuff, I mentioned some potential manufacturing variables. These are some of the specific ones that they look at, like the FDA would look at. These here are some of the most important steps in the manufacturing process. So, what method are you using to weigh your active ingredient and your excipients? Um, how are you ensuring that it's accurate? Calibration of your scales, believe it or not, and things like that. We're getting down to that kind of level of detail. So, blending uniformity. Believe it or not, and you might have learned this in pharmaceutics, but believe it or not, if you don't use the appropriate blending method to blend your active ingredient with the excipients, you may get non-uniformity, which is a huge problem, especially for a drug that has a low therapeutic index. That means if you just dose it a little higher than you're supposed to, you have severe toxicity. So things like that, you absolutely have to have uniformity. So you want to know that every tablet has whatever it says. If it's 100 milligrams, you don't want it to be fluctuating more than I'm going to say 5%, but they do, and I don't know the specific number, but they do have kind of a regulatory value, that it's allowed to deviate, but it has to be uniform is the idea. And different blending methods will be necessary for different formulations. Um, they have to do an active pharmaceutical ingredient release test. How fast is it released from the drug product? And how do you ensure sterility for parenterals? And that parenteral almost always means injectable, but technically it means anything that's administered outside of the gastrointestinal system. But really, most people just mean injectable. The therapeutic objective influences the product design. Does the ailment require rapid or chronic drug action? If we require very rapid drug action, of course, we're not going to have a slow release formulation, right? So the formulation has to be determined based on what we need. So, if we're talking about chest pain or angina, of course, we don't want a slow release formulation. So they use a sublingual formulation that doesn't even have to get swallowed because it's absorbed in the capillary system under the tongue, right? So that formulation has to be designed very carefully. So it can't be just kind of random powder excipients. It can't be just a big blob of cellulose with some nitroglycerin in there, which would be the active ingredient, would be the nitroglycerin. It's got to be carefully designed, and that's been done for decades now for nitroglycerin. But that's the example, a good example there. Does the ailment require local versus systemic drug action? If we want it to work in the colon, and we're gonna call that local because the colon is not inside the bloodstream. If you're talking about if you have like an ulcer in the colon or something, for example, how do we ensure that the drug gets to the colon and not it doesn't get absorbed and go through the whole bloodstream? How do we target it to the colon? There are ways to do that with biopharmaceutics. There are ways to add excipients that prevent disintegration or dissolution until it reaches the colon. So, yeah, I mean, all these things are really, really critical. So with the active pharmaceutical ingredient, they do solubility and dissolution rate tests. Okay, the physical and chemical properties determine the dissolution rate and the solubility of the active pharmaceutical ingredient. Okay, so it's important to understand how fast it's dissolved, which is the dissolution rate, and the solubility. And these are different, and I want you to understand very clearly. I want you to understand the difference between these two. The dissolution rate is the speed at which the drug dissolves. The solubility is once it reaches equilibrium, what is the final amount that is dissolved? What is the total amount that's dissolved rather than the rate? Okay, so solubility is not, those are not identical. Solubility and dissolution are not identical terms, particularly dissolution rate. So we need to know those for each active pharmaceutical ingredient. And of course, really we're looking at the whole tablet or the whole formulation dissolving and seeing how fast we release the API. And the solubility is important because we may not want a drug to be soluble in one portion of the GI tract, but we may want it to be soluble in another portion, for example. And there are many other examples where we might want different solubility levels depending on the purpose of the drug. And I don't want to get into all those right now, but we can later on with different dosage forms. Crystalline versus amorphous. So why are we talking about that? This seems very specific, right? It's actually very important because drugs that are crystalline or in their crystal form um, will dissolve at a different rate than drugs that are in their amorphous form. And generally, amorphous drugs, if we wanted to talk about this from a physical pharmacy perspective, amorphous drugs are generally more rapid dissolving. They have basically higher free energy to dissociate, amorphous drugs. Okay, so crystalline drugs are more stable and they take longer to dissolve. So why does that matter? Well, it actually can affect the onset of action of a drug. Okay, so you can't just, in a company, you can't just randomly, you synthesize the active ingredient and maybe it's pure, but then, you need to know, is it crystal, what crystal form is it? Is it crystal form A, B, C, because there are different shapes, different crystal forms, or is it amorphous, which means no crystallinity? And how does that affect the solubility or dissolution rate? Because it'll affect both. Particle size determines dissolution rate. Okay, so you can't just, again, in a drug company, they can't just kind of put a random array of different active pharmaceutical ingredient particles in their formulation and hope for the best. They have to confirm or ensure that their particle sizes are uniform and that they meet a predetermined size that they aim for, that has been proven to be absorbed at the right rate and extent for the therapeutic indication that they're looking for, that they're looking to target. The salt form of the active pharmaceutical ingredient is, in many cases, is relevant. Okay, and hopefully you remember a little bit about this from chemistry, but the salt form is because most drugs on the market they do have charged groups, either negatively charged or positively charged groups, which will usually be again a carboxylic acid or an amine. Well, they have to be formulated with the conjugate acid or base, depending on which one we're talking about. So, for instance, the first one I could think of, because this appears to have some clinical relevance, is metoprolol succinate, succinate. That is a salt form, and that, believe it or not, sometimes you can't just say, oh, the drug of choice for this disease is metoprolol. You actually have to specify it's metoprolol succinate or metoprolol tartrate. The salt form, in many cases, only one salt form might have been thoroughly tested in a clinical trial and proven to be effective. And we may not always know for sure, is that salt form really better? But sometimes only one form has clinical evidence, so it's safer to go with that. So it's proven that, of course, that the salt form can determine how fast something is absorbed because of how fast it is dissolved. So these are just examples. Basic drugs might be formulated with citrate, succinate, tartrate, the hydrochloride salt, or another salt of an acid. Acidic drugs may be formulated as the salt of a base, such as sodium hydroxide, tromethamine, etc. So when you see all these weird terms after the drug itself, those are just the salts, and they're sometimes they're important. Sometimes they actually make a difference in that drug performance, that drug product performance. And sometimes these salts have their own biological effects, which we try to avoid. Right? We try to avoid that as much as possible. Once in a while, they might have a biological effect. The salt form is generally used to enhance solubility, but the counterions pKa may affect drug absorption. Okay, and don't worry about that right now until we get to the next few slides, and then later on in the actual, like the full biopharmaceutics lecture, you will see a lot more. So this is just a basic demonstration, or pretty much an image to show you how the salt form of a drug is produced between again an active pharmaceutical ingredient and the conjugate. So, for instance, if we just have an active pharmaceutical ingredient, we have to ionize it. We have the counter ion, which could be an acid or base depending on the drug. That would be like the succinate or the tartrate or the citrate. That will be ionized. We kind of combine them together, and you would do that in a solution, for example, dissolve them together, and then maybe supersaturated, or you're going to maybe evaporate that solution, and you're going to form a bond then between the positive and negative charge, the two different species, and then you're going to have crystallization, and then that's just a drug salt. So it's pretty simple. But many drugs are administered as a salt form, where they have to be because they're charged. Um, Again, many of them are, and in most cases, the salt form of a drug makes them more soluble and faster dissolving. Now, and let me go back a step. Saying that the drug has to be administered in the salt form is not always true. Some drugs can be administered as the free base or the free acid. That, for example, if something is a carboxylic acid drug, if it has a carboxylic acid functional group, if you leave the proton on, so it's COOH, then that's called the free acid. Okay, and that means if there's the hydrogen or the proton on there, that means it's not in the salt form at this point. So you can administer drugs that way, that they usually don't dissolve as rapidly, and that's the main difference, I would say. So this slide is just showing how, for the same active ingredient, how if it's combined with different salt forms, with with these different counter ions, you could basically get a different ionization profile depending on the pH. And later on I'll get into how this might look on the effect on absorption. We go into the full biopharmaceutics lecture, but this is just kind of the introduction to that. So I don't want to go into too much detail. But for example, in this graph, if we look at the kind of green diamonds, that would be that's the actual active ingredient, which is Clavix or clopidogrel, and this is the percent ionization. I know it's hard to see, so I apologize, but that's ionization. So we're at 100% at the lower pHs, okay, and that would mean to me that if an acid pH makes it ionized, it's most likely it's got to be a weak base, right? Because weak bases, the amines generally receive an extra proton or ionized at low pH. Okay, so that would mean that the conjugate ion should be a salt or an acid, and that would be, for example, bisulfate or bisulate. So first of all, getting back to the green diamond, we're going as the pH rises. Just imagine this might be going from, for instance, the stomach and closer to the maybe the small intestine. As the pH rises, the ionization goes down. Okay. Well, I mean, so that you can definitely guess as to how that might affect drug absorption. It's never smart to guess, right? They have to test that. They just have to test the different conditions to see what will happen, rather than just kind of assuming we can, make an accurate estimation. But if you had to guess, I would say that, as you get closer to this pH 6, because less of it is ionized, that means more of it is neutral, and that's the active ingredient. I would say that it would actually be absorbed faster. So why would I say it's absorbed faster? And the reason for that is because your plasma membrane is lipid, and there's evidence. I mean, decades and decades, or maybe more, maybe centuries of evidence that the more fat soluble or lipophilic a chemical, the quicker it is absorbed. Okay, so as we make it less and less ionized, it will absorb faster. There's always a problem, though. Potential problem with that is if we make it so ionized, or so unionized, or so nonpolar that it doesn't dissolve, that it cannot be absorbed if there's zero dissolution. So I guess the point is this is kind of complicated. So what we have to do, what the drug manufacturers have to do, is find a happy medium where we still have good dissolution, we still have good absorption, and that might be somewhere, who knows, somewhere in the middle, something like that. So, but again, not getting into too much more detail on absorption until later on. But I also want you to at least make note of this other thing, these other curves, and that's the bisulfate, which is the triangle, and then the bisulate. So if we change the salt type for that drug, if it's clopidogrel bisulfate or clopidogrel benzoate, we actually get that the salt will have a different ionization at the same pH. So just say, for instance, pH 2. Well, pH 2, we have more ionization of the bisulfate. That means the bisulfate might be quicker to dissolve in the stomach than the benzoate. And that might matter, right? And I don't want to, again, I don't want to go into any further predictions without getting to that until we, to get to that further lecture. But that, without, again, too much more discussion, just saying that it does matter. Yes, it will matter. I want you to understand that it does make a difference in the drug dissolution and absorption. Okay. Again, and if you want to get into specifics, again, with the pHs over here, pH 6, clopidogrel will mostly be unionized, enhancing absorption, pH 5. Some will be ionized, and some of it will be, some of it will be ionized at pH 5. Not very much, right? Maybe 70% or something will be ionized at pH 5. When it's ionized, that will allow it to rebind to the counter ion, which is the either benzoate or the bisulfate. It'll allow it to rebind to the counter ion, which is 100% ionized at this pH. And then the other question is, when it's bound to the counter ion, is it prohibited from being absorbed, or is it absorbed quicker? Well, that's going to depend on the drug. So again, we don't want to make any assumptions. It's important to understand that all these things matter with each different drug. So another consideration in biopharmaceutics is the route of administration. Oral is mostly limited to small, stable molecules. If our goal, again, that's if our goal is the typical goal, which is systemic absorption or absorption into the bloodstream, which is most oral drug products. Generally limited to small, stable molecules. So usually not a protein, right? And everybody's trying to develop ways to administer proteins orally with systemic action, but for the most part, that's almost impossible. Topical, okay, must typically be formulated to cross the stratum corneum. Now, topical doesn't have to mean just on your skin. Topical could mean anything really that's not going to be systemic. A lot of those can be topical, right? It could be could be anal administration that's not absorbed, right? It could be, some people might call, like if you're treating something in the esophagus, you might call that topical. You don't have to, I guess. But essentially it means it's not getting absorbed in the systemic circulation, and it's treating like a local target. If we are using it, which is a standard topical, would be just on the skin, must typically be formulated to cross the stratum corneum. Right? Usually we want it to cross that's the dead skin. So it has to be lipophilic enough to do that. Parenteral means apart from the GI tract, but again, it's usually injectables. And that's really all, because I already mentioned that before, but I just want you to understand what parenteral means in practical use. In the real world, usually means injectable, but technically, if you want it to be very technical, it means apart from the GI tract. That would be the route of administration. Transdermal means through the dermis layer of the skin. Transdermal is not really topical. It's not, because transdermal means you go all the way through the dermis layer of the skin, and the intent is generally systemic absorption or systemic exposure all the way into the bloodstream. So that's why you have transdermal patches for pain, like Duragesic, or that was the brand name, but fentanyl patches and other patches are right. They're not meant to just treat that region of skin. They're meant to get absorbed into the systemic circulation. That's very different from topical. Inhalational, usually used to target the lung, but it can lead to systemic exposure. Okay, that's usually not the goal, right? Usually the goal is just to treat the lung, something in the lungs, like inflammation, or I think there are now some new examples of antibiotics that are inhalational, but mostly we're talking about things like asthma or COPD and things like that. Another consideration is the dosage form. Um, there are different types of tablets and capsules. These are just some general examples, and there are much more specific examples we can get into, but there are immediate release. And let me go back a step. I know a lot of this is going to be discussed in just your pharmaceutics course itself. So there's going to be some overlap because this is biopharmaceutics. But immediate release and these are very specific terms that you need to be familiar with because they're going to come up over and over again in the curriculum. So immediate release means dissolves. It's most drugs that you have, they dissolve as early as possible. So if you go to the store and buy naproxen or ibuprofen or something like that for pain, they're typically they're immediate release or they dissolve as quickly as possible. Extended release means it still starts to dissolve early, but it continues for a long time, right? So extended means it does have an action still early on, but it's a longer and slower action. Okay, so it might last all day rather than just for a few hours. Delayed release is very different. I would say it's different from extended release because it means for some period of time there is zero drug release, so zero API release from the formulation. So it might be an hour, it might be 2 hours, but it starts dissolving late, either to avoid the stomach acid or to target, for instance, to target the colon. So enteric coated, that is really part of the delayed release type of formulation. That's one type of delayed release. Okay, but and that would be specifically to avoid the stomach, to avoid dissolution in the stomach. But there are delayed releases that don't dissolve at all until they get closer to the colon. So that's a very specific instance. Parenteral dosage forms, there are many out there, but some of them are here. Suspension depot form. That's a powder suspension, usually for intramuscular administration. So they inject it right into the muscle tissue. Okay, and of course it'll kind of slosh around or mix around in the interstitial fluid there in the muscle. But because it is injected as a suspension, right, that means most of it is in a solid or powder form. That means it's going to be a very slow release and long release. And that could be really useful for something if you want very long-term activity, long duration of action. It might be useful to have a depot form like that. And the reason why it is a very slow dissolution is because there's very little mixing in that fluid in the muscle. Very little mixing going on at at any time compared to like the GI tract. Okay, so it's a kind of like a sink effect where as you pull some out, your body pulls some of the active ingredient out of the muscle and puts it into the blood, and then more of it dissolves. And then the cycle continues. So you keep getting more and more from that powder kind of dissolving slowly. The PEG form, or polyethylene glycol form, is similar, but in many cases it might already be totally dissolved, but it's sometimes used intramuscularly, and the polyethylene glycol slows the release. So the PEG makes it a larger molecule, and it could make the IM injection be a longer duration of action because it's slow release. And then there's solutions for injection, and that means it's already completely dissolved, right? Just a simple solution. Could be an antibiotic, could be anything, right? But usually we're talking about kind of a hospital setting or an inpatient setting. But solution for injection is one of the few forms that can be used directly into the veins, right? Direct IV use. You wouldn't, it's not considered safe to inject any undissolved powder right into the vascular system, right into the veins. It should already be dissolved if you're going to do that. Other dosage forms would be patches, creams, lotion, gel, ointment. So these are usually topical. At least the cream, lotion, gel, and ointment are usually topical. Patches can be, but it all depends. They could also be intended for transdermal. But patches have a solid matrix to release the drug. Obviously, right, because they have to have something to hold it. something to hold on to and kind of adhere to your skin. So there's a solid matrix there to release the drug. Lotions, creams, gels, ointments have a semi-solid vehicle to release the drug. And without getting into too much detail, the makeup of those, um, the inactive ingredients in those formulations are actually pretty important with respect to how fast the drug will get absorbed. Um, so again, the matrix vehicle will give different drug absorption characteristics based on the hydrophilic, lipophilic nature of the vehicle and the drug. So if your vehicle is hydrophilic but your drug is hydrophobic, you might repel that vehicle might repel the drug and push it into the skin. Whereas if the vehicle and the drug are hydrophilic, it may be very slow absorption because the skin is more hydrophobic. So the main point is that um, it's really important to test these things and to determine which is the best combination of hydrophilicity or lipophilicity of your vehicle and your drug. Implants, they became popular probably when I was a student for contraception, but there might be many other types nowadays. But a subcutaneous implant, there are many different types of things, but they have generally a very long, slow release rate. It might be a month, it might be weeks, might be several months actually, right? So that's a very kind of unique scenario where you have an actual like solid with the drug inside of it that would slowly release the drug over time. So lastly would be the excipients. And I want to kind of clarify that we talked about salt forms of drugs, and the salt, although the salt is not really the active ingredient, right? The salt is not at all. So we talked about clopidogrel, what was it, bisulfate. Those salts are a little different from an excipient because they are chemically bound with the active ingredient. So they're technically not really excipients, where the real excipients are the inactive ingredients that are not really not chemically bound to the drug. So those would be things like cellulose, talc, lactose, and there are many purposes. One of the purposes for excipients are just to increase the mass of the dosage form. So the easiest example is imagine if any of you have ever worked in a community pharmacy, you've probably dispensed some form of levothyroxine sodium. I know when I was a student, I can't even tell you how many different brands there were in generic forms, but it was like Levothyroid, Armour Thyroid, Synthroid, levothyroxine sodium. It's pretty much all the same thing. It was thyroid hormone, but they were all in microgram doses, right? So I wouldn't expect you to know this offhand, but as a scientist, measuring micrograms is extremely difficult. And if you just gave someone the active ingredient and were somehow able to press it into a tablet form with just a pure active ingredient, these things would be minuscule, probably almost invisible to some people. So the whole point is, sometimes you have to add something else to make it a practical, convenient size for the patient to consume. And that's why they are still pretty small, right? Because they don't want to make them bigger than necessary. So they're still really small. But these are these multicolored tablets that you'll find at and sometimes a physician might write accidentally a milligram strength, but they don't mean that, or at least I hope they don't. But these, basically, that's probably the extreme example where the excipient, we need a lot of excipient just to make up the mass. There are other examples. I personally, in an experiment we were doing, we had to measure or weigh the just acetaminophen or Tylenol tablets, and I think it said the strength was 350 milligrams, and we weighed them against that's 350 milligrams of acetaminophen. We weighed them, and each tablet was like 360 milligrams. So that meant almost 100% of it was active ingredient. It might have been 10 milligrams of binder or some excipient. So it all depends on the active ingredient, right? Not everything has to have a lot of excipient. Some excipients are there to promote favorable rheological properties during production. If you don't remember that, rheology is the study of the flow of matter. And when you're producing a drug right before it is in its final formulation, it has to be very easy to pour. It has to flow well. It has to be easy to mix. And sometimes the excipients are necessary to promote that flowability, right? Some are there to make bioavailability consistent. As I mentioned, you might have to, for instance, protect the drug in some cases from the stomach acid or from intestinal enzymes or something like that. But essentially just to make the bioavailability consistent. Changing excipients can significantly alter drug stability and absorption and uniformity. Okay, so it's not something that's done lightly. And if a drug company comes up with a formulation that works well, it is not if they ever wanted to change it, it's not something you do overnight, right? If they ever had a reason to try to change it, if they ran out, for instance, they ran out of an excipient, if there was a shortage in the world of lactose or something, it's not an easy switch or a quick switch to change the formulation, right? Because the FDA requires them to test every single aspect of that drug again once they make a minor change to its formulation. And I have a quote on the bottom that I won't read, but that's from the AmericanPharmaceuticalReview.com website, which is kind of a nice website if you're interested in this area of pharmacy. So kind of to summarize, biopharmaceutics involves factors that influence product design and manufacture, stability of the API in the product, release characteristics of the API from the product, availability and release of the drug at the absorption site, delivery to site of pharmacological action, such as colon or systemic, and many of these are influenced by differences in physiology from patient to patient, as well as concomitant medication or food consumption. I'll have one more slide on biopharmaceutics, and then I'm going to transition over to the introduction to pharmacokinetics. So the ways we can test biopharmaceutics, and again, this is just the introduction, and we have a longer lecture on this later, coming fairly soon. But the ways we can test biopharmaceutics, there are several ways. There are in silico ways, in vitro, and in vivo. And the method that a manufacturer will use depends on the drug, right? It depends on the disease they're treating. So the FDA might allow, believe it or not, the FDA in some kind of rare cases, they might allow a company that's producing a generic drug to just use computer simulation or computer-based methods, which is called in silico, to determine if their product will meet the requirements for bioequivalence to the brand name product. And that's pretty rare, but it can happen. So in silico, again, we can sometimes predict the absorption based on calculated log P or the octanol water partition coefficient and solubility of the API. Again, that's rarely accepted alone by the FDA, but we can just use computer-based models to determine, what would the solubility be of this API in this formulation. In vitro is very, very commonly used. The FDA in some cases will allow you to grow cells in the lab. They're used as a barrier to measure drug absorption. One of the most common cell types in history for this kind of test is the KCO2 cells, which are from a colon cancer cell line. And I've grown them once in my career, but it's kind of a slow-growing cell line, and what they do is they grow them until they reach something called confluence, meaning there are no gaps between the cells. And the cells are normally suspended on a membrane, and there's a fluid compartment above and below, and then they would expose them to a drug on one side and see how fast does the drug go through, so they can replicate through these colon cancer cells, kind of replicate or predict what would happen, hopefully predict well what would happen inside a real body with the colon or the small intestine. Right, so that's in vitro there, and that's in many cases that's well accepted by the FDA. Another method is to remove animal intestinal tissue to provide a more realistic barrier. So that would be probably a rodent in many cases, but removing the intestinal barrier, putting it putting it in a device that kind of separates one side from the other side with fluid, and then adding a drug product to see what happens and how fast. You might add like a whole, the whole powder might already be dissolved. It all depends on your purposes. In vivo, preclinical animal studies are used, but ultimately human trials are required for new drugs and to some extent for generic drugs. So yeah, they can use, and I don't know, to be honest, to the extent that they still use them, but for many years they use beagles, beagle dogs, in these types of pharmacokinetic tests, and I believe they still do to some extent. And it's not really for toxicology testing; it's more for absorption and things, just pharmacokinetics testing. And sometimes, believe it or not, they have beagles or dogs that will be in a company for like their whole lifespan, and their purpose is to be, to utilize them for these types of pharmacokinetic tests. So all the way from in silico to in vitro to in vivo. Okay, and what the company, it's usually, you start off with a brand name company, they're making the brand name product, and then they have to do these things. But then if you're ever going to replicate it as a generic company, you don't necessarily have to repeat every single one of these things. It all depends on the drug, the everything about it. What is the therapeutic index of the drug? How important is it to have absolutely identical profiles and things like that. Okay, now we're going to start the introduction to pharmacokinetics. Pharmacokinetics is a science of the kinetics of drug absorption, distribution, and elimination. If you look on the bottom here, that's really, these are all the processes kind of summarized. And what it really is we want to quantitatively describe each portion of this. So absorption, distribution, and elimination. If we could develop equations and constants that can describe those uniquely for each drug, then we can predict what would happen for each patient, try to predict what will happen based on the dose and the frequency that we give a drug. It involves experimental data collection, which then forms the basis for mathematical model development. So pharmacokinetics does require experimentation first, and then we start to refine models that can quantitatively describe how that drug is, well, really the disposition of the drug throughout the whole body. Software is becoming more and more critical. In fact, it's used ubiquitously now for pharmacokinetic studies. And statistics, of course, are critical in PK studies in drug companies, the FDA, and in academia. Pharmacodynamics is the study of the biochemical and physiological effects of drugs on the body. When I say pharmacodynamics, I generally just mean the mechanism of action of a drug. Okay. Pharmacology is more vague. Okay, I pretty much use pharmacology and pharmacodynamics interchangeably, but many other healthcare professions may not use pharmacology the same way. They may just mean in general just pharmacy. Okay, so it all depends on who you're talking to, but pharmacodynamics is really not. There's no gray area there. That means the drug mechanism. It's also describing the dose-response relationships between a drug, and so when a drug is administered, or even in vitro when a drug is used, what is the biological response to that drug? And you develop graphs or curves. Therapeutic receptor target versus toxic receptor target. Pharmacodynamics, in that field is important to delineate the difference. So some drugs have different targets which produce toxicity than the therapeutic target. In other cases, the therapeutic target might also produce some toxicity, so it all depends on the situation. So that has to be determined, for each drug on a kind of a unique basis. This is actually a major focus of PHA 421, and then in all pharmacotherapy modules, you will really learn about pharmacodynamics of each different drug class. Clinical pharmacokinetics is the application of PK to drug therapy in patient care. So we have to consider a lot of things that wouldn't normally be considered when you're running a kind of a healthy population study. So we're talking about patients' disease states, their organ function changes, age, sex, concomitant drugs. So clinical pharmacokinetics is taking the fundamental pharmacokinetics and applying that in a real patient scenario. So in this slide, I decided to actually discuss the drug monograph or package insert, and I did that because I think in many cases throughout, and it's probably in many pharmacy schools, probably throughout a lot of curricula, there's not a lot of time spent on the package insert. However, the package insert is just a wealth of information. When you're out there actually working in a pharmacy, you can pull off that little pamphlet or drug monograph or package insert where you pull that off the bottle. Sometimes, even though you have electronic databases, you have your cell phone and things, sometimes the information that you need is easiest to just get it right there, because it's right there in front of you, and it has pretty much almost anything you could need for that medication. So on first glance, it can be confusing with all the terminology in a really large package insert. However, the one kind of comforting piece of knowledge is that every single package insert has to have sections organized the same way. And if you immediately, if you know that you want to know something about the pharmacology, clinical pharmacology of a drug, which, by the way, pharmacokinetics, at least on the package insert, pharmacokinetics is listed under that as a subsection of clinical pharmacology, you would know to go to section 12, and then in many cases it might be 12.3, to find the pharmacokinetics of a drug. Okay, so everything you could think of is going to be in that insert pretty much. And again, if you specifically wanted to know anything about the kinetics, you would go to that section. Again, that's the 12th section. Unfortunately, the earlier sections, sometimes, I don't know if it has it here, but a lot of times the earlier sections are more about warnings, right? Warnings about toxicity and things like that, and allergies and things like that. Whereas, as you can see, that is number 5, but you'll even see that usually in the beginning of the package insert. Whereas the part that I'm interested in, which is the pharmacology and even the kinetics, comes usually always much later on in the section 12. So from a practical perspective, yeah, we can start in many cases with the package insert. For instance, when a physician determines the dosage regimen, or if the pharmacist wants to verify that we're starting with the correct dosage regimen, you can utilize the package insert, which is usually, right, it's now in many different electronic databases that you would use with your cell phone or your computer in your work setting. But sometimes it's not going to be enough, and sometimes we're going to have to measure the blood concentrations of a drug, and that's where the clinical pharmacokinetics really kind of has this kind of like moment of fame. And this is usually required for drugs that have a narrow therapeutic index, or it's very easy to to reach a toxic level with certain drugs. So usually you'll measure their concentration in an internal medicine type of setting or hospital setting. Um, so we right. So one patient, you never know. In one patient, you might be giving them the normal dosage regimen, and they might be the normal weight and everything, apparently normal, but then they might reach a toxic concentration, whereas no other patient did. So I guess a couple questions we can ask are, what can cause an abnormal toxic response to a normal regimen? Okay, and I think I'll just answer that now, but later on in the lectures, I will have a lot of questions that I want you to try to answer on your own. But what can cause an abnormal toxic response to a normal regimen? Okay, and so one of the things that can happen is that the patient might have, even if their organ function appears normal, right? Normal kidney function, normal liver function, they may have a mutation in, for instance, in a transporter or a metabolic enzyme, right? So that's pharmacogenomics. They may have a mutation that you won't see that on just a standard organ function test. And that, what if that mutation was important, right? That enzyme that is mutated or that transporter that is mutated is important for elimination of the drug. And then in many cases you'll see excess drug in the bloodstream and then toxicity. And that's not something you can predict without kind of getting the genotype of that patient ahead of time. What can cause low therapeutic effectiveness with a normal regimen? Same idea. It could be the drug is absorbed poorly. It could be due to a transporter that is mutated, or that it could be overexpressed. For instance, that P-glycoprotein or MDR1, and we're going to talk more about that later. But that's an efflux transporter that in some patients is overexpressed, and it reduces the bioavailability of a drug. So in the real world, a lot of things don't go according to plan in terms of pharmacokinetics. And the best thing you can one of the best things you could do first is determine in those cases, at least is the concentration of the drug where we want it to be. Is it in the normal range? And if the patient still doesn't have the appropriate response, you have to start considering other things like receptor downregulation or receptor mutations. Even if the drug is the right concentration and they're not getting an appropriate response, there may be other reasons. There's going to be other reasons that may not be directly related to pharmacokinetics, right? Maybe related to the pharmacodynamics of the drug, and that might be different in that specific patient. So that's when you're left with, your pharmacology knowledge that you'll be learning about as well. Your pharmacology knowledge might tell you, well, maybe we should choose a drug with a different mechanism, for example, because we're reaching the right concentration, but the drug just isn't working, right? So it's always going to be a combination of your critical thinking and the guidelines, right? So a few definitions here. One of them is drug exposure. And drug exposure is the dose and measurement of the drug concentrations in the plasma or other fluids over time. So I want you to become very familiar with these terms because you'll see them many times in my material and later on in the clinical material. Cmax is the maximum concentration of the drug usually in the plasma. Okay, Cmin is the minimum concentration of the drug in the plasma. CSS is the steady state, which is once the drug reaches a steady concentration. What is that concentration? And then area under the curve, and that is a measure of the total drug exposure. And we're going to talk about that. I don't want to define it now, but we're going to talk about that as we proceed with the next few slides. Drug response is a measure of the pharmacological effect of the drug. Receptor occupancy, that's not clinical, usually measured in vitro. Surrogate markers of effectiveness might be blood pressure, lipids, cardiac output. Remember, well, I don't know how much you've talked about this yet in any class, but these things are usually considered secondary endpoints in most clinical trials, because none of these things are, so the patient doesn't necessarily see these things. And what I mean by that is the things that are really the desired endpoint is survival for a longer period of time than so basically extending someone's lifespan, or reducing mortality, improving their quality of life, whereas these are just means to that end, right? But those are markers of drug response. Okay, and these are sometimes really useful to know. Again, like I said, the desired clinical endpoint is the final drug response, which is things like reduced death or reduced mortality, or at least extension of life. Toxicokinetics and clinical toxicology, which we won't spend much time on after this, those are still very important, and those are subfields. But toxicokinetics is a study of the pharmacokinetics in drug overdose. What I think is very important, I do have a toxicology elective, right? But of course, not everyone will take that. So I wanted to emphasize, at least here, some of the most important things in toxicokinetics, and that is, not the first bullet. Of course, we don't do that in humans. We don't we don't intentionally study toxicokinetics in humans, unless there's a large number of humans who overdosed, and then we can maybe gather some data. But normally it's tested in animals. But the most important point is that second bullet. Pharmacokinetic constants are usually completely invalid in an overdose situation. Right? Pharmacokinetic constants are usually completely invalid in an overdose situation. So if I tell you the half-life, elimination half-life of a drug is 8 hours, well, you may not know what that means yet, and we're going to talk about that. But half-life is just the time it takes for 50% of the drug concentration to, basically the drug concentration decline by 50% in the plasma, how long that takes. However, half-life, in many overdose situations, there is no half-life because the drug is not eliminated in a first-order manner or in a half-life manner. It's eliminated in a different manner because of saturation of elimination routes. So what I want you to understand is that once we get into toxicology, a lot of the pharmacokinetics that you find in a package insert are not going to be applicable to that at all. Okay, so you're in a whole different ballgame then. So I guess that raises one question. So even though we haven't talked a lot about half-life, what is required in order for a drug to have a true half-life? Okay, and again, this might be really tough if you haven't thought about it before, but what it is that, by definition, right, a half-life is the drug declines by 50% in one half-life. So if you think about that, that's 50%. So just say we start with 100 micrograms per milliliter concentration. 50% will be 50. Another 50% would be 25, and then be 12.5. So you're not eliminating the same mass per unit time. You're eliminating the same percent per unit time. And that's strictly because you have not saturated your organ systems. That means your liver or your kidney or both, they still have additional spare capacity to eliminate more drug if you increase the concentration of drug. Okay, so what's required is that we have not saturated our elimination or excretory routes, and it's required that we eliminate a consistent percent per unit time and not a consistent mass per unit time. And I will delve much deeper into that as we proceed throughout the course to show you what that looks like. Clinical toxicology involves the identification and treatment of poison patients, of course. That's a subspecialty that pharmacists can become credentialed in, right? You could be board certified in clinical toxicology. That would be what DABAT, diplomat of the American Board of Applied Toxicology. You can get that credential. We've had students ultimately end up doing toxicology fellowships and things like that. So it's kind of an interesting kind of off the beaten path type of field. So throughout this course, we'll be talking about drug concentrations. So first point here of order is how do we measure them? Okay, you won't need to know how to use any of these things, but at least one exposure is probably worthwhile. So drug concentrations may be measured in various biological fluids. Most commonly, though, rather than any kind of old-fashioned method, they're usually using things like gas or liquid chromatography, more rapid methods. You might have seen these on any one of those TV shows where they are doing kind of forensics and things like that, or crime scene investigations. But gas or liquid chromatography, something that I use in the lab quite often actually is a UHPLC or mass spectrometry, and usually these things are combined. So GCMS or liquid chromatography MS or mass spectrometry. And that allows you to take a very complicated mixture such as plasma and prepare it very, very little preparation. Just take the plasma, maybe get rid of the protein, run it through a machine, and then quantify your drug. And most of the work is done by the instruments, not by the personnel. So example fluids that we would want to measure drug in, of course, would be blood or plasma, spinal fluid, synovial fluid, right, in the joints, that is, urine, saliva, feces, or rarely what you're exhaling. We don't usually do that in pharmacology, but maybe in toxicology, of course, we might be looking at the exhaled air Um, of course, we're all familiar probably with the breathalyzer, or have heard of the breathalyzer test for ethanol. So this slide is just a little bit of a focus, more of a focus on drug concentrations in the most common fluid that we use, which is blood, plasma, or serum. Serum or plasma is the most commonly used fluid for pharmacokinetic analysis. Technically, there is a difference. Serum is obtained by allowing the whole blood to clot and then taking the clear supernatant. And that means serum is devoid of or lacking the clotting factors. It's a little bit of a difference there. Plasma is obtained from supernatant of blood that has been anticoagulated with heparin or ethylene diamine tetraacetic acid, or EDTA. You've probably, well, if you work in a hospital, you might have seen the vacutainer tubes that have EDTA, or if you've ever done any kind of phlebotomy work or something like that. That's to prevent the coagulation of the blood. Plasma, in that case, it still has the clotting factors because you've prevented coagulation. Okay, so there might be a subtle difference in drug concentration because the plasma would have still have more stuff in it, right? It would still have the clotting factors, so that might have a bearing on the drug concentration, in the end, if you haven't removed those clotting factors. Maybe a small effect. Fluid in blood is in equilibrium with all tissues, so that's why it's the most commonly used fluid for pharmacokinetics. Because the blood is where the drug usually is first widely distributed, and then it's in equilibrium with every tissue, like the brain, kidney, liver, etc., etc. So that's why we use it most often. Some drugs are bound to plasma protein. It could be 0 to 100% protein bound, and that's really, really important to understand. And we're going to talk, when we talk about drug distribution, we're going to have kind of a long discussion on plasma protein binding. So it's critical to understand if you're getting lab values on a patient. And of course, to be honest, most of this is already standardized, so you shouldn't have to guess. But it's still important to understand if the lab is reporting the total drug or just the drug that is not bound to plasma protein. Because the analytical methods that I talked about, you have the option, right? The analytical method could be designed to measure total drug, so that's bound and unbound, or just the free drug that's not bound to plasma protein or not stuck on that protein. So it's important to understand that difference. If free drug is measured, but the drug is protein bound, it is possible to underestimate the amount of drug in the blood. Obviously, right? So if I claim that the drug is only at a concentration of 10 micrograms per milliliter, but I didn't know that they were only reporting the free drug, if 90% of that drug is bound to protein, then I'm, have approximately a 90% underestimation of the concentration in the blood, right? So that's really important to know what is being measured. So throughout pharmacokinetics, we're going to see these curves, usually a plasma level versus time curve. Okay, and the plasma level just means plasma concentration. The PK analysis requires knowledge of drug concentration over time. I mean, the entire lifeblood of pharmacokinetics is the ability to plot something and then generate some kind of mathematical relationship or equation based on that. The MAC is the minimum effective concentration. The MTC is the minimum toxic concentration. Okay, and honestly, in some references, you might see slightly different abbreviations used. So you just need to be sure, whenever you see one of these, just be sure you know what it means in whatever you're reading. But MTC-MEC ratio is a therapeutic index. Okay, the higher the safer. Now, in the other course in PHA 421, I talk about a more specific definition, which is LD50 over ED50. But this is just kind of a general way of looking at that. Basically saying we want the effective dose to be tiny and the toxic dose to be gigantic. And what that means is that when we are treating a patient, if we've achieved a therapeutic effect, we want to be nowhere near the toxic dose, right? We want that toxic dose to be astronomically high. And that is true for some drugs. It's pretty rare, but like maybe some antibiotics are hard to achieve toxicity with. Whereas other drugs, if you get the dose wrong by, if you double it, for instance, you might have some serious toxicity. So it all depends on the drug. So typically you want to be in the therapeutic window. That is above the minimum effective concentration, but below the minimum toxic concentration. And so the reason why they call it a window is on a graph like this, you can draw kind of a square. It will be like a window. Okay. So, however, there are cases where we might allow a medication to drop below the MEC, or the minimum effective concentration. Okay, and I just wanted to see if you pause it right now, pause it for a minute or two and think about that. So I'm going to answer that question the best I can now. So go ahead and pause. Okay, so what that is the reason why that might happen sometimes is if a drug is toxic. For example, if the drug is toxic at the same level that it's therapeutic at, for instance, cancer drugs, anti-cancer drugs, unfortunately, there may be no window here, no real window, where the toxicity is separated from the efficacy or the effective dose. That effective concentration might go hand in hand with some toxicity, so you might have to let it drop below the effective concentration for that patient to recover before you retreat them. So, in some cases that is necessary. There are other examples with antibiotics where it's been proven that it's more important to achieve a high peak concentration than it is to achieve a long duration above the minimum effective concentration. So it's a time versus peak concentration that's important. And so it all depends on the drug. Okay, so sometimes we might not have to stay above the MEC all the time. Okay, so there are many, many other terms that we'll go over throughout this course. So PK analysis includes a large number of other terms. Some of them are, as I already alluded to, Cmax would be, for example, the peak concentration. Tmax would be the time at which you achieve the peak concentration. So the Tmax is the time at which you achieve the Cmax. So whatever time we're at the highest blood concentration, that's called the Tmax. The area under the curve is if we could integrate this curve and quantify the entire area. You can think of it as almost a surface area in a sense. If this were like a flat object like it is, kind of like the surface area under this curve tells us the total drug exposure. And that allows us, believe it or not, to calculate a lot of other things later on that we'll get into. And so they'll be discussed in much greater detail for each administration route throughout the course. So I want you to start becoming familiar with these basic terms because I'll be repeating them and using them many, many times later on. So some of these, the Cmax and Tmax are, used all the time. AUC is used quite often. So please become those. So we were talking about drug concentration in the plasma. What about drug concentrations in other tissues? What's critical to understand is that the plasma concentration of a drug may not accurately reflect the concentration in another tissue. What I mean by that is so this is plasma, what if you measure the plasma concentration of a drug? We just get this hypothetical number, 20 micrograms per milliliter. But what if I looked in the kidney cell? Literally, if I had the ability to look inside of the kidney, individual cells, those epithelial cells in the nephrons, what if it were at 100 micrograms per milliliter? Believe it or not, that can easily happen. So it's never good enough to just look at the plasma concentration and assume you know everything about that drug in the body or where it's distributed. In some cases, a lot of it could be concentrated in one tissue or another tissue, and that's mostly because of the transporters in each tissue. The kidney has transporters that can concentrate drugs, concentrate them much higher than their plasma concentration, and that can happen in the liver as well and some other tissues. So that's the critical point, is that you can't just rely on plasma concentration to understand what's happening to the tissues. And again, in this example, that could potentially lead to selective renal toxicity or selective pharmacological action or pharmacodynamics in the kidney. So the toxicity one is kind of interesting to me because sometimes you'll see a drug or a chemical that is only toxic to one organ, and then you look at that organ and you find that it's super concentrated there. So the organ may not have an inherent susceptibility to it. It may just have a higher concentration of that drug. Measure kind of drug concentrations might be in excretory fluids. The urine is a commonly used fluid, and one of the main purposes would be to determine how effective is the kidney at eliminating this drug. And so essentially what we'll calculate is something called the renal clearance. The renal clearance, I'm not going to define it now like specifically, but it's just a measure of how strongly or how well that kidney can eliminate the drug. And in many cases, the best way to determine that is to measure the urine concentration of the drug and the plasma concentration of the drug and see how much is extracted through the kidney or by the kidney. Fecal concentrations can reflect unabsorbed drug or a drug that has undergone hepatic, biliary, intestinal transit, and that means a drug can get absorbed, right? It can still get absorbed, but then as soon as it reaches the liver through the portal vein, it might get pumped back out through the bile, might get pumped right back out into the duodenum, right? So if a drug appears in the feces, it either wasn't absorbed at all, or it was recycled from the liver, portal circulation, liver, bile. Right, those are the two options. Or in some cases, we'll find metabolites in the feces, and that usually indicates, of course, the drug has gone through the liver before reaching the feces. Right, we can figure that out. So I guess the important points here is that we can find drug in the feces either because it wasn't absorbed, or it may have actually gotten somewhere in the body. Could be the blood, it could be just the portal vein itself, but it could be systemic blood. But the drug still has the opportunity to get back into the feces in some cases. Okay, every approved drug has mass balance studies reported. So if you look at the package insert, you should see this in pretty much every approved drug I've seen has this reported. So when they administer the drug to a real patient population, what percent of the drug is eliminated through the feces? What percent of the drug is eliminated through the urine? Okay, and what metabolites are produced, and how are they eliminated? So anything you need to know about that drug, usually you can find it in the package insert. Again, with respect to what organs are most important for eliminating that drug. Usually they use a radio label. That means they might use tritium or C14. So they'll have someone synthesize the drug in a radioactive, usually low toxicity, right, almost harmless radioactive form, and then they could very easily study through using a scintillation counter. They can actually study where that drug, how it's eliminated and where it ends up. So they could literally, like, if they were using it, for example, if they were using an animal, they could take each organ and run it through a scintillation counter to quantify the total radioactivity, and that'll tell you the exact percent of that drug that ended up in that tissue, the drug or a metabolite of the drug, right? Because the radioactivity won't tell you what form it's in; it'll just tell you that is there somewhere. That the radioactivity is there in some form or another. So why are drug concentrations sometimes not enough? So why could 5 people have the exact same drug concentration in the plasma, but totally different therapeutic responses? And I want to highlight this right now, and that's the brackets I have around drug here. Okay, I won't use this every time, but I'm going to use this from time to time. That's kind of like a shorthand way of saying concentration. So drug concentration in the plasma. So why could 5 people have the exact same drug concentration but totally different therapeutic responses? And the answer to that is because there are differences in physiology, right? So even if they have the same pharmacokinetics, they may have different receptor sensitivity to the drug, and right, and I mean that's the main point, right? They may have different receptor sensitivity. They may have mutations in a receptor that reduce the activity of that drug. Okay, so the whole point is that you can't just rely on pharmacokinetics, as the endpoint is saying, okay, then the drug is going to be effective in this patient because it reached the right concentration. You still have to measure the markers of effectiveness. For example, if it's a blood pressure or like a beta blocker drug to lower heart rate, you can't just measure the concentration of the drug. You have to measure the heart rate and things like that. That's why, again, monitoring the drug concentration is usually combined with monitoring markers of effectiveness or toxicity. And the reason why I have markers of liver damage there is because just like the effectiveness of the drug can differ in multiple patients, even at the same drug concentration in the blood, the toxicity could differ as well. Someone might have a more sensitive liver, and you may not have identified that until you gave the drug, right? So you still have to monitor all these things. You can't just monitor the drug concentration. Even bacterial cultures in two patients given the same antibiotic at the same dose with the same infection, maybe one person has poor tissue penetration of the antibiotic, even if they have the same blood concentration. Maybe there's something unique about the organ where the infection is, where they're not getting enough tissue penetration. Again, even if they have the same blood concentration. And that sounds far-fetched, but that can happen in some cases. In many cases, one good example is meningitis. That I always think of that example, and in people that have meningitis or inflammation of the meninges in the brain, the ones that have the higher inflammation, it's been shown that they generally get more drug distribution into the brain. So you can get a higher drug absorption of the brain if you have higher inflammation in many cases. So that may not be reflected in the blood concentration, because even if the brain has twice as much drug in one patient, because it's kind of a smaller volume, you may not see a large difference in the blood concentration, right? They may not be reflected in the blood. So now we're going to move on to pharmacokinetic models. We're going to cover them briefly here, and then in the subsequent lectures I'll talk in much more detail about the pharmacokinetic models. So they're mathematical models, and they're used to design drug therapy regimens during clinical trials, and they could also be used to predict drug exposure post-marketing. So a model is just a way of representing mathematically what your best guess or your best estimation of what is happening to the drug in the body pharmacokinetically. So it's a hypothesis using mathematical terms to describe quantitative relationships concisely. Models are developed by first generating experimental data. So you have to give the drug to a patient or a subset of patients, right? A small number. Monitor the blood concentration over time, and then start generating these the data for all these different terms. We've talked about Cmax, Tmax, area under the curve. We've talked about half-life. You get all these parameters. You get the value, like an average, right? And then you can kind of like use that to basically generate the model. And when we generate the blood concentration versus time curve for all these patients, you get data points on the curve, but then you actually have to fit a curve to the data using an equation. Okay, so the curve has several mathematical constants or parameters that define it, and I just mentioned those, some of those, and one of them is like the half-life is one constant that should be associated with each drug in the normal therapeutic range. An example use of a pharmacokinetic model might be if I wanted to know the drug concentration in the liver 1 hour after oral administration of a 20 milligram dose, and usually you'd include things like the patient weight. So it gives you the ability to predict something before you do it, right? So that's the power in the pharmacokinetic models. If we can get one that works well, we can get a really good idea of how much medication to give a certain patient population or even an individual patient, with just a few parameters or few bits of information about that. PK models can be used to predict drug levels, calculate optimum dosage regimen, estimate accumulation of drugs and metabolites in the blood and in other tissues, evaluate the differences in rate or extent of availability between formulation. That would mean bioequivalence. Describe how differences in physiology or disease states affect ADME, and ADME is the absorption, distribution, metabolism, and excretion, or predict drug interactions and their severity. So there are many, many different types of pharmacokinetic models. There are the classic models, which we'll spend a lot of time talking about, and then there are the more advanced models, which would try to incorporate data, kind of kinetic data from each single tissue. And we won't spend too much time on those because they're better left to the software, and the complicated equations are better left again to utilizing the sophisticated programs that are out there. So the model types, there are different types. So compartment-based. Compartment-based means it does not use data on drug movement or metabolism in individual tissues or organs. So if I'm going to develop a compartment-based model, I'm usually just measuring the blood concentration or the plasma concentration of a drug over time. I'm usually not measuring every tissue, every drug concentration in each tissue. So compartment modeling does not intentionally take into account individual organs in the math, but rather looks at drug distribution as having multiple phases. So this is going to be important as we proceed, and you're going to get a lot more on this, so don't be too concerned if you don't understand it yet. But when we are doing compartment modeling, a compartment is really actually defined as it's really a different phase. If we look at the curve of blood concentration over time, it represents a different phase of that curve, and that different phase likely reflects distribution at a different rate in different tissues. And I know that may not make a lot of sense right now, but I think it will as we proceed. If you look at that third bullet, hopefully it helps a little bit. For some drugs, the whole body may receive drug at the same time and rate. And it's that simple. That would be considered a one compartment model. So for example, if I inject a drug into somebody, IV, into their veins, right, and then it distributes almost immediately, right? So the blood concentration, it spikes to the top, to the peak almost immediately, and then it goes down. And the way it goes down, the way it declines, is predictable and consistent, and there's no inflection, right? The curve is consistent throughout. That generally means that we have a one compartment model. That means it distributed immediately, and there's only one distribution phase. However, there are drugs that distribute into the blood immediately, but then into, for instance, two different organs at the same rate, and then another organ at a different rate. And this is just one example. So what this is saying is that if a drug goes into the blood immediately, but then it goes into the, another organ slower, and then another organ at a different rate, every one of those different rate constants for each distribution phase, that's going to be considered a different compartment. What's important for everyone to understand is that even though it's tempting to consider a compartment an individual organ, pharmacokinetically they're not really defined that way. And I'll give you an easy example. For example, like if a drug is distributing into two different organs at the same pace or same exact rate, we'll bunch those organs together and call that a single compartment. Okay, so it doesn't have to be an individual organ. A compartment could be a cluster of tissues and organs that happen to have the same rate of distribution. Okay, so that's why there's one compartment models, there's two compartment, there's three compartment. Usually we don't have to go beyond two compartments to get a good pharmacokinetic model for a drug. The simplest compartment model is a single distribution phase, or one compartment. The compartments are always represented by some kind of box, for instance, and this would be the only compartment, so compartment 1. Now the kinetic constants that are associated with this would be, kind of a bare minimum. We always need an absorption constant and an elimination constant. Now I have K here. You see later in the course, I'll start to refer to as K subscript EL for K elimination. Those are really interchangeable, so please don't be too concerned about that. Either K or KEL, I mean elimination rate constant. So what are these? What is now? You guys know what constants are. I mean, you've seen constants in a lot of courses. But what are these rate constants? What does it actually mean? And to really define it, it actually means, first of all, they're associated with their own units. So I have to tell you the Ka. I can't just say Ka. I could tell you the Ka in whatever units we're using to fully describe it. So if I say Ka per hour, right, that actually means the fraction of whatever drug is available, whatever drug is present. In this case, if we're talking about oral absorption, whatever drug is dissolved and available in the GI tract, that would be that drug times the Ka, which the Ka is the fraction of drug absorbed per unit time. And if I say per hour, that means the fraction per hour. Same thing with the elimination rate constant, the K or KEL. Whatever drug concentration we have in this compartment, and we're usually talking about the blood as where we're measuring it at least, the concentration of that compartment times the KEL will give us, that's the fraction, right? The KEL is the fraction of drug eliminated. And if I say per hour, if that's the K that I'm giving you and it's per hour, that means the fraction eliminated per hour. So you would take the concentration of drug in the plasma, multiply by the KEL, and that would tell you how much is going to be eliminated, or the rate at which is going to be eliminated in that given moment of time. Now the K's do not change, okay, but the concentrations in the compartments change. So if the concentration in the plasma goes down, and then you multiply by that by the K, that gives you the mass eliminated per unit time, and that's going to decline over time, right? So as your blood concentration declines, the mass eliminated per unit time also declines, because this is considered a first order rate constant. So first order rate constant, that always means that a specific percent is eliminated per unit time, not a specific mass, right? So we're not going to say 10 milligrams every hour. We'll say 10% every hour, or whatever percent it is. Every drug is going to be different, right? An elimination doesn't have to mean leaving the body. It may mean degradation through metabolism. So if a drug gets destroyed or metabolized in an organ, and that metabolite is not active at all, then that's effectively eliminating the drug, right? It's doing the same thing. So in some cases, or theoretically, an enzyme in the liver and renal excretion. So let's kind of break this apart. An enzyme in the liver would do metabolism. Renal excretion is not metabolism, but it is drug elimination, right? It's going into the urine. If those both happen at the same rate, that would be reflected in one single K value. Okay, that means both of those would be reflected in that K value. So the K for elimination, or the KEL, is going to be the combination or culmination of all the different routes of elimination at the same time. And notice how this model is not defining, so what organs are involved. That's because all we're doing to develop a one compartment model is monitoring the blood concentration. So we don't necessarily define where the drug is going, we just say that it is going, and this is the rate at which it is going, and we just know, though, because of the nature of the body, we know that the K, or the elimination rate constant, is a combination of multiple different elimination routes in some cases, right, at the same time. So we can break this into different models. So there's one called a one compartment IV. Notice the Ka is gone. There's no absorption constant, because one compartment IV is, the assumption is that because it's IV, 100% of the drug is reaching the compartment almost immediately, and there's no absorption rate, because the absorption rate is only dependent on how fast you inject the drug, which is so much faster than oral absorption that we consider it instantaneous almost. So the only thing we have is one compartment and then an elimination rate constant. Two compartment IV, if we get rid of the absorption phase because it's IV, we have the first compartment, but then look at this. That second compartment, guess what? It has its own rate constants. One of them is the rate going in, or the drug going into the compartment. One is the rate going out of the compartment. And then we still have the K, or the KEL, which is the elimination rate constant. So now we're getting more complicated here. And if I wanted to know, for instance, let's see, so how much drug, what is the rate of drug appearance, or how much drug is appearing in compartment 2 per unit time, I would have to know the concentration of drug in 1 and the current concentration of drug in compartment 2 and these two rate constants. And I'm going to show you how you would basically how you generate the equation for that, what that would look like. We can go a little more complicated, and then again, we do one compartment. We already actually I showed this in the last slide. One compartment, and that was the assumption was that was oral because we did have a Ka. So that's what that looked like. But then if we went with two compartment PO or per oral, that would have the Ka. It would have compartment 1. It would have compartment 2. It would have each kinetic constant for that compartment and an elimination constant. And these could get dramatically more complex if, for instance, compartment 2 could also eliminate. Then there might be a second KEL or elimination constant. Okay, so I really don't have you guys doing a large number of calculations with these compartment, well, at least with these models here, at least for the first few lectures. And there will be a few selected calculations I will have you do later on, though. But please understand what the K's mean. Each K really stands for the fraction of drug present in the compartment. The fraction of that drug present that will be moving to the other compartment for whatever unit of time I utilize. So K2 1, K2 to 1 is K compartment 2 to compartment 1. That's the rate at which we're removing the drug from compartment 2 and placing it into compartment 1. And that always requires you to know the concentration in each compartment. Another model type that I'm only going to bring up just now, I don't think we'll really get into this in the other lectures because, again, the math involved is going to be a lot more sophisticated and it usually requires software. But this other model type is a physiologically based model, and that is, in the grand scheme of time, from the decades of pharmacokinetics existence, PBPK models are pretty new. And what they do is they attempt to incorporate drug transport and metabolism in every single tissue pertinent to a drug's distribution in the body. So what it is the equation involves every single piece of knowledge that we have about every tissue. So there will be rate constants for the kidney, rate constants for the liver, and each one of those might have subconstants which might reflect each transporter in each organ. So we're not going to get into that too much because it's very mathematically complex, or at least there are a lot more constants involved. It could be really, really critical, or I should say really, really useful, if we wanted to know exactly how much drug is in a given organ at a given time. We would have to use something closer to this rather than using a compartment model. Again, PBPK models are not considered compartment models. They're a different type. The only way you can ever develop one of these models is when drug distribution in every tissue is known. And that is very difficult to determine in real patients, because you'd have to do biopsies and things like that. Now, animals can be used to try our best to develop a PBPK model, but of course, the organ systems of the animals are going to differ to some extent, so it may not be perfect. And this is an example, and you're not really going to be responsible for telling me what all these things mean for PBPK models, but I just wanted to give you this one example in case any of you end up in a field where you're doing pharmacokinetics. In PBPK models, major organs are handled separately. Other slowly and rapidly equilibrating tissues have separate constants. Okay, so the heart is handled separately from the kidney, from the liver, and if we start on, well, we'll start on the right side here. This is the arterial blood, and each one of these Q's is the flow rate, the rate of blood flow to each organ, to the heart, muscle, kidney, liver. And the SET and the RET are kind of undefined tissues that are slow eliminating tissues and slow equilibrating tissues and rapidly equilibrating tissues. Okay, every single one of these will have its own K constant. The Km in the liver means the K metabolism, so the metabolic rate. The Ke in the liver is in this case, it means the renal elimination rate. And we would have so on and so forth for every organ, so you can predict the concentration of the drug in the blood over time. Okay, so without going into too much detail, one of the advantages is if we can somehow develop a PBPK model, I'm not just looking at blood. I'm not just actually modeling the blood. You can use the equation to predict how much drug is in the kidney per unit time, how much drug is in the liver per unit time, and how much drug is in other tissues like the fat or adipose tissue. And that could theoretically be really useful if the target were in one of those tissues. If we had a target in adipose tissue, just imagine a hypothetical drug that's used to cause lipolysis or fat loss. Maybe it would be helpful to have a kinetic model, PBPK model, to predict that. But for the purposes of testing, I just want you to understand, with respect to all the models, the PBPK model, which is physiologically based pharmacokinetic modeling, the main purpose is to predict what's happening rather than just in the blood. It's to predict where the drug is and what concentration it's at, and really in every tissue involved. That is much more detailed. So in summary, or to summarize, drug product performance is considered the release of the drug from the product and bioavailability of the active pharmaceutical ingredient. Biopharmaceutics provides a scientific basis for drug product design and performance by examining the interrelationship of the physical chemical properties of the drug, the product, and the route of administration on the rate and extent of drug absorption. Pharmacokinetics is the science of the kinetics of absorption, distribution, metabolism, and elimination. And really, elimination is pretty much made up of metabolism and excretion. The plasma drug concentration versus time profile provides data from which various pharmacokinetic models can be developed, and the purpose of models is to allow predictions to be made to aid in choosing dosing regimens. So I'll just wrap up the lecture with some learning assessment and application questions. At this point, I'll be revealing all the answers, so you can choose to kind of pause it, and if you haven't already tried to answer them on your own, please do that. I really encourage you to do that. So I'll give you a chance now to pause it and do that, and then I'll be starting any second now. Okay, so what I'll be doing for some of these is just telling you the answer, and for the other ones, I'll actually be revealing it on the slide. So please be prepared to write a little bit on these. So first of all, what section of in the package insert will you find pharmacokinetics? The answer to that is section. We'll just say the clinical pharmacology, which is section 12. I won't ask you the subsection, but it's 12.3. So section 12 is pharmacokinetics. What is the significance of the plasma level time curve? Okay, and there's not really like a verbatim answer to this or a black and white answer, but the significance of the plasma level time curve is that it tells us the drug exposure, the total drug exposure, and then the drug exposure over time, and that allows us to determine if we're in the therapeutic window for the patient. How does the curve relate to the pharmacologic activity of the drug? Well, it's important to understand that while the plasma concentration is related to the the amount of activity of the drug on its target is not a perfect predictor of that. It's important to understand that sometimes multiple patients could have the same drug concentration but have different responses. And in some cases, the plasma concentration is not reflective of the concentration of the drug in the tissue that you're trying to target. What is the purpose of pharmacokinetic models? The purpose is to describe mathematically how the drug disposition occurs, or the drug disposition in the body is, and that is the main purpose is to be able to make calculations to predict the drug concentration at a given time in a patient population. And then I'd like to draw a diagram describing a 3-compartment model. I think we've only covered 2-compartment models, but with your knowledge now, you should be able to draw a diagram describing a 3-compartment model with first-order absorption and drug elimination from compartment 1. Okay, so we're moving on to the next question. This model represents drug eliminated by renal excretion. Okay, right here, biliary excretion and metabolism. The drug is also distributed into compartment 2 from compartment 1, and then vice versa from 2 to 1, so there's an equilibrium here. And then the drug is also metabolized, and the Ku stands for renal excretion of those metabolites. So there are 3 compartments, but the question is, how would you determine the overall rate constant for drug elimination from compartment 1? And I want to clarify, I don't mean distribution, so we're going to forget about the K12 and K21 right now. Really the question is, how would you determine the overall rate constant for elimination? So the real elimination routes are when the drug is either leaving the body or getting destroyed. So that would be renal excretion, biliary excretion, and then hopefully should go out through the feces actually to be eliminated, and then metabolism to be destroyed. And this is a lot simpler than you might have expected. So all you need to know to generate one of these is you need to know how to add these constants together. And believe it or not, it's not as complex as one might expect. So the total elimination constant, or KEL, believe it or not, is just adding the biliary one, the metabolism constant, and the Ke, which is, in this case, it means renal excretion. If you add all these, because remember, these are fractions. What fraction of the existing drug in compartment 1, what fraction of it is eliminated per unit time? So because in no case should these all add up to more than 1, right? They can't add up to more than 1 because they're just fractions, right? Fractions of 1, and they have to add up to something lower than 100% per unit time. Well, in most cases, I should say, they're going to add up to less than that. So KB, Km, and Ke, add those together. Imagine if that were like 0.2, 0.2, 0.2, that would be 0.6. And the overall rate constant for elimination would be, it should be larger than each individual constant. And that would mean 0.6. Again, hypothetically, if it's 0.6, that means 60% of the drug will be eliminated per unit time, and I have to define that. And if I say these Ks are based on per hour, that'd be 60% per hour. And where it gets complicated, and you'll see later on, is that every minute of that hour, the drug concentration will decline. So remember that we're no longer starting off, for each minute of time, we're starting off with a different concentration, and then the Ks are still the same, right? But when you basically calculate the total mass of drug removed, it's going to decline also per unit time. So the more drug you start with, the faster you get rid of it by mass. So as the further it declines, the smaller the mass is going to get, because it's based on percent of that mass, or fraction of that mass. So adding a little bit more complexity to this, that kind of question. So write an expression describing the rate of change of drug concentration in compartment 1. So you would write dC1 over dt. That means change in drug concentration in compartment 1 over change in time would equal what? How do we calculate that? Again, it's a little more complicated. So what we would do is say, first of all, we have to account for two things. We have to account for drug leaving compartment 1 and then drug entering compartment 1. Remember, drug leaving compartment 1, and now the question is not, it is not just elimination. I'm saying in one way or another, the drug is leaving compartment 1. It doesn't have to be just pure elimination. It could be distribution. So we're going to have to utilize KE, KB, KM, and K1 to 2. Remember, all those arrows are, look at that, all those arrows are leaving compartment 1. So all those constants should be able to be combined. And we're saying that we're going to take the concentration of the drug in compartment 1 and then multiply it by each of these constants added together, right? And that would be one part of the equation, and that's how fast is the drug leaving 1. On the other hand, some of it's going to be entering 1 through distribution from compartment 2. So we'd have to take the drug concentration in compartment 2 and multiply that by K21. And what we get is this equation. So the change in drug concentration in compartment 1 over time, it's going to be, first of all, let's look at the right side. It's going to be negatively affected by all these 4 elimination or at least routes where drug is leaving compartment 1, right? KE, KB, KM, K1, 2. And every one of these constants, every one of these reflect drug leaving compartment 1. So we take all these together, and that whatever that fraction adds up to together, then we multiply by that by the concentration of drug in compartment 1. But then what's going to contribute positively and add drug to compartment 1 would be, if we're accumulating drug from compartment 2, is diffusing or entering into compartment 1 using the kinetic constant K2 to 1. So we take the drug concentration compartment 2, multiply by that by K21. We put those together, we get the ultimate answer. Is drug leaving or entering compartment 1? Now, I want you to think about this a little further and make sure it makes sense to you guys. So if you overloaded compartment 2, whatever that compartment is, it could be multiple organs. If you overloaded them with drug, I don't know how, but however you can do it, if you inject it into there, right, eventually you should be able to force, even if drug is leaving compartment 1 by these routes, you should be able to force a lot of it in through distribution back into compartment 1. So we should be able to overwhelm these elimination routes and still gain more drug over time in compartment 1, and that will be represented by this. If we did C2 really, really high, this negative would become less and less relevant if we had a very large amount of drug in compartment 2, and that constant doesn't change because it is a constant. So C2 times K21 might be massive. So ultimately, the drug concentration compartment 1 would be a positive number it would be a positive change. So it would be increasing the concentration compartment 1 over time. And that's how all the software that's out there that's doing these kinetic calculations, it's all based on these constants and things. And they're not, if you break it down, they're not super complicated, right? At first it may look overwhelming, but it's not absolutely terrible if you think about it enough and digest it. Okay, more of a theoretical question, conceptual rather than calculating. So consider 2 biologic compartments separated by a biologic membrane. Drug A is found in compartment 1 and in compartment 2 in a concentration of C1 and C2 respectively. So we have 2 different concentrations. So there's different concentration in each compartment. Answer the following questions discussing physical-chemical properties of the drug and properties of the compartments that might influence equilibrium conditions. So what possible conditions or situations would result in the concentration C1 greater than C2 at equilibrium? Okay, so I want you guys to, again, so pause it now, 5 seconds, and then I'll continue. 5, 4, 3, 2, 1. Okay. So what possible conditions or situations would result in concentration C1 greater than C2 at equilibrium? And if you think about it, if we just had if you had like a beaker with a membrane in between, the concentrations on both sides, if there are pores there in the membrane, the concentration should be identical on both sides of that membrane. So what I'm saying here is that in a biological tissue, it's possible for one compartment to have a higher concentration than another compartment. Okay, and in this case, we could think of them as different tissues, even though compartments pharmacokinetically it may be multiple tissues together, but we could think of them as single tissues for this example. In one of the answers, I mean, you could think of a lot of things probably, but one of the answers would be, for instance, if there are drug transporters that would be forcing the drug into compartment 1, right, more rapidly than into compartment 2. So if it's not just simple diffusion, if the drug is being transported into the compartment 1 and concentrated there, that's one reason. There are other reasons that we could talk about, and we'll get into this later on in the course, but what if in compartment 1, what if the pH of that compartment made the drug more ionic or more charged and prevented it from moving into compartment 2? Whereas what if in compartment 2 the pH made the drug neutral, so then it could diffuse freely into compartment 1? So the pHs could be different. There could be transporters, like I said, first, pumping the drug into one compartment out of the other one. And then there are probably a lot of other things you might be able to think of. But those are the two main ideas I wanted you to consider. Under what conditions would C1 equal C2 at equilibrium? That would be if there's no transport, either the pHs in both compartments are identical, or the pKa of the drug is so different. In fact, it might be so different from the pHs of those solutions that it doesn't even matter if they're different. So if the pKa of the drug is really high or really low, and it's in the pHs of those compartments have no bearing on the ionization state of that drug, perhaps the pHs of the compartments may not matter. So the main idea there is that if the C1 and C2 concentrations are identical for a single drug, these two different tissues or compartments, it might mean that the amount of drug ionization is the same in both compartments, or the nonpolar percent versus the polar percent is not different in the two compartments. And again, if there were no transporters that handled that drug, and if it were just simple diffusion, then that's another reason why they could be the same concentration. And one other example that people use a lot is the brain concentration of a lot of drugs is lower than the plasma concentration. That's partly because it takes a long time for the drug to get into the brain, but also and that's because of the barrier, but also partly because sometimes there are drug transporters that pump drugs out of the brain to keep them in the blood. So it's a little complicated. So thirdly, the total amount of drug A in each biologic compartment, we're going to call that A1 and A2, and that means the total mass, not the concentration, but grams or milligrams of drug in compartment 1 is A1 and compartment 2 is A2 respectively. So describe a condition in which A1 is greater than A2, but C1 is equal to C2 at equilibrium. And it's not as bad as you might think. Okay, so that means the total mass of drug in compartment 1 is higher than the total mass of drug in compartment 2, but the concentrations are the same. And that's actually really easy, because the only way that would usually happen, right, or the only way I could think of is that compartment 1 would have a higher volume, right? If compartment 1 has a much higher volume, even if it has the same concentration, of course there's going to be more total drug there, right? Because if it's 10 liters in compartment 1 and 1 liter in compartment 2, with the same exact concentration, there's more drug mass in compartment 1. So please think about these things and make sure they make sense to you.